Hi, my name is Danny, and I did my project over the last 8 weeks over critical congenital heart disease and the Notch1 gene. So, what is critical congenital heart disease? So this is a group of heart defects that are within the congenital heart disease group that occur before birth during the development of the heart in the fetus. Now, what makes this more critical than typical regular congenital heart diseases is this impacts the infant's survival rate after birth by affecting the blood oxygen levels. Now, a lot of these different heart defects that I have here really affect the amount of oxygenated blood that is pumped throughout the system compared to the deoxygenated blood. I'm probably going to really botch the way I say these for the 7 main heart defects, but the first one is trancus arteriosus, transposition of the great arteries, tetralogy of Fallot, pulmonary atresia with ventricular intact septum, tricuspid atresia, hypoplastic left heart syndrome, and total anomalous pulmonary venous return. Now, I have some pictures over these next few slides about these different critical congenital heart diseases and defects that happen within these fetuses and how we really can see why the deoxygenated blood is overriding the oxygenated blood and causing this to become a critical condition for these infants, and that surgery is required within the first few hours of life for them to survive. Obviously in this one we can see that this vein or artery and vein is connected to the aorta, and that deoxygenated blood is being passed into the aorta and throughout the whole rest of the body system. There is this septal defect in both of these that affects the blood being pushed appropriately either through the left or right ventricles. Also with tetralogy of Fallot, we're seeing this as well, that deoxygenated blood is being able to seep into that left ventricle and go to the aorta when it should be being pushed into the lungs to be oxygenated. Now this is that major venous switch where the aorta is actually attached to the right ventricle, so the blood that is being pumped into the body and throughout the body is deoxygenated blood, which is obviously not what our body needs to be able to continue to survive. And this arterial switch operation is what needs to be done to be able to help this infant survive and have the appropriate amount of blood supply, blood oxygen supply to the rest of their body. And then this is that hypoplastic left ventricle. Obviously, compared to the size of the right ventricle, there is not enough space for that left ventricle to hold the appropriate amount of blood that we should be seeing to pump to the body. It is causing a lot of problems where the aorta is actually attached to the right ventricle instead of the left ventricle. And then this is the total anomalous pulmonary venous return, where these pulmonary veins are coming back to that left atrium to be able to be pushed through the aorta after the blood is being oxygenated, but instead of returning there, it is going back into that left ventricle, and so that back into the right ventricle, right atrium, right ventricle instead of the left, and then that deoxygenated blood is going in through the aorta. Now, what causes this? So a lot of things with certain and other types of congenital heart diseases. There's many different types of genes that scientists have tried to locate within these different defects that happen between these fetuses of why there is this genetic reason for these happenings. So I have listed here a lot of different mutations and genes that are related to the development of the heart that are on different chromosomes and how they potentially could have their effect on the development of the heart and these congenital heart diseases. Now, I focused on specifically Notch1 on chromosome 9, but we'll get into that in just a moment. Now, typically these heart conditions do arise alone, but they can occur with other syndromes. Typically, we do see heart disease problems with Down syndromes, Turner syndromes, and the 22q11.2 deletion syndrome. A lot of these syndromes do have other metabolic problems as well associated with them, because a lot of those other genes and the chromosomes that are being affected are being seen also with these syndromes. So it's just an added thing that on top of those that they have to deal with. But as I stated, they really don't know what's kind of going on. Most of the time it can be genetic cause with some of those genes causing it, or it could be a sporadic event. In most of the cases that they have studied, between the chromosomal abnormalities, these nucleotide microdeletions, or adding of a nucleotide base, that's only in about 10 to 15% of cases that they come across when they are able to test them genetically. With the infants in familial cases, if they're looking at specific families, about 1 in every 9 infant, has this problem going on, and they can actually identify it genetically and not a sporadic event. Now, with families that have no history of heart problems at all, and the infant ends up having one of these congenital heart diseases, also known as critical congenital heart disease as well, but these mutations that occur are new, so de novo, which is a new mutation that has happened. This also has occurred within about 10 to 15% of the cases of these patients, but that doesn't account for the rest of the 70% of cases that are there. A lot of the scientists do think that these effects are environmental, by the prenatal care that is happening for the mothers, the area that they're living in, exposure to carcinogens during the specific development of the heart, during the specific time for the development of the heart. So it's really important that obviously, even if you don't have a history of heart problems in your family, being able to understand, the environment that you're in can also have an effect on some of the development of the child. Now, into Notch1. So this is the protein formation of the Notch1 protein in the gene. Now, this is actually considered an autosomal dominant gene. So if a child were to receive, say, an A allele, a large A allele, while a lowercase a allele, they would have this Notch1 protein developed with the potential to develop into one of these congenital heart diseases. Now, some of these act alone per case, and some of them have also all these other genes interacting with each other that cause these defects within the heart. Now, the Notch1 receptor protein, this actually does a lot for the cell. It does the cell determination of what it's going to be. Like, is it going to be a liver cell, an epithelial cell, a heart cell? And it helps with the growth of the cell, maturation, and it also helps with the signaling of apoptosis. Now, with this, when it does have a mutation, it can be considered an oncogene and be related to cancers as well. There are a lot of different oncogenes that we see are related along with congenital heart diseases, with this being one of them. For mutation-wise, what happens with this protein, say in a heterozygous individual, there's a nonsense mutation or there is a single base pair deletion. Now, obviously, these can happen as individual events, either during the development of the fetus or as a genetic cause passed down from either the mother or the father. Now, specifically, one of the this Notch1 gene does affect the aorta from some of the research as well, and it also is known to affect what is known as aortic ventral disease 1. And as you can see here within my pedigree, you can really see that this is obviously an autosomal dominant gene, unfortunately, I have that this father has passed, and he has passed this on to his children. Now, he is heterozygous, while the mother is homozygous recessive. So some of the children end up not having this disease and have a life without being affected by any kind of congenital heart disease, or a risk of potentially cancer in the future. Now, there's a lot of genetic testings out there that you can do. I know we've talked recently a lot about the direct-to-consumer genetic testings. Now, personally having a lot of history of heart disease myself, I've always thought it would be interesting, but I don't think with how sensitive some of these things can be and how critical they can become when you are in the process of trying to grow a child or a fetus, it can become a problem. There is a genetic company in Wisconsin called Prevention Genetics that does offer two different congenital heart disease panel. There's the comprehensive and the non-syndromic. They test obviously a different amount of genes. The Notch gene is included in them along with the other genes that I listed at the beginning of this presentation. Now, early detection to help these infants is really important. Now, parents can be tested to understand if they carry this, but also there's a lot of testing that parents can do as the fetus is growing within them. They can start it with a simple blood test and go all the way to an amniocentesis. Each one has their own risks of knowledge and potentially hurting the infant as well. Usually having a conversation with your doctor about those procedures and diagnostic tests before you decide to have that done is recommended. For the infants themselves, genetic testing is recommended, but it is not required. Though there is a law that each newborn is required to be tested with a pulse oximeter to measure their oxygen levels. This is really important, especially with those main 7 critical congenital heart diseases that I briefly touched on, because those are obviously going to really affect the amount of oxygenated blood within your system. This is going to be picked up by that pulse oximeter and going to warn medical professionals, when they have a lower oxygen level, like more diagnostic testing is going to be needed, and that, this might not be a healthy fetus. Unfortunately, though, for cost-wise with these genetic testings, they do have pretty penny to them compared to some of the direct-to-consumer genetic testing ones there are out there. For the 2 above that I have listed, they do cost between $1,000 to $1,500. And here's my references.